Alrighty, our last organic molecule that we will be covering in this chapter is nucleic acids. And nucleic acids, again, are just ginormous molecules, but we're going to try to at least try to understand how these molecules are made up. This time today, we're actually going to start with the chart with organic molecules on it, and we're going to fill that out first. So, nucleic acids have three pieces to their building blocks, and I have them color coordinated so I can try to explain them to you when I show you how they're structured. There is a backbone for the nucleic acid that's made of sugars and phosphates. And then to that sugar phosphate backbone, we're going to add a nitrogenous base. The purpose for nucleic acids is to store and pass on genetic information. So whenever any type of cell reproduces, it's got to pass on all of its genetics and characteristics to its offspring, because that contains all the information for how that new organism is going to function, how it's going to work, how it's going to appear, all the characteristics that it has. Okay. There are two specific classes of nucleic acids that we will be spending a lot of time in the coming up chapters talking about. One is DNA and the other is RNA. Now DNA, again, you've probably heard of DNA before. It actually stands for deoxyribonucleic acid. The double helix form of DNA is very familiar to us. We've seen it in books before, and it kind of looks like a ladder. Now, the diagram that's in your book was printed kind of small, and so that's why I also gave you a coloring sheet to go with this, because you really need to understand how a DNA molecule is put together, because that's how you're going to understand later on how cells reproduce. So even though it's going to be a little bit before we get back to this again, I want to make sure you understand the structure of a DNA molecule. And so when we get done with this here today, after you do your other homework, take some time to really work with that coloring sheet, use different colors for different pieces of it to make sure that you understand how this DNA molecule is hooked together. A DNA molecule looks a lot like a ladder. It's probably the best way to think of it. And the long pieces, the side arms of the ladder, are made of your sugar and your phosphate. So for DNA, it's going to be what's called deoxyribose sugar. And if you had RNA, it's a ribose sugar. It's just a specific type of sugar that we've talked about when we talked about carbohydrates. Okay, so you've got a deoxyribose sugar that's linked to a phosphate, that's linked to another sugar, that's linked to another phosphate, and this is going to be really, 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 really long, but I just have a small portion of the ladder on my whiteboard. Okay, then the other rung of the ladder is going to be the same, a deoxyribose sugar, and then a phosphate, and then a sugar, and then a phosphate, and then a sugar, and then a phosphate, and then a sugar, and then a phosphate. It's going to keep on going like that. Okay, now the rungs of the ladder are made up of a nitrogenous base, and there are four specific kinds of nitrogenous bases that are used in DNA. You have adenine, you have thymine, you have guanine, and you have cytosine. So those four are the only four that are going to be used in DNA. RNA uses three of those and then one that's different. And again, we'll get into some of those differences a little bit later on. What's crucial here, though, is that your bases are going to attach to the deoxyribose sugars. And in order to try to simplify this drawing a little bit, I'm going to use an A for adenine, a T for thymine, a G for guanine, and a C for cytosine. So one of my nitrogenous bases is going to attach on this side to the sugar, and let's say that I attach an adenine over here, okay? And then let's say that when I get down to this sugar, let's say that I attach a guanine right there. And as I continue going on down this particular side, I might attach another adenine, and then a thymine, and then a cytosine, and then an adenine. And so I'm going to have this long, long, long string of all these nitrogenous bases attached. This side is going to be very similar. But in order for these guys to hook together, what is the case is that adenine only pairs with thymine, and that guanine only pairs with cytosine. Okay, so an adenine can't hook to a cytosine, it can't hook to a guanine, it can only hook to a thymine. And then what happens is that this adenine and this thymine will actually hook together via a hydrogen bond. And we talked about hydrogen bonds extensively when we talked about the characteristics of water. So it's the same type of bond that's in with water molecules, but this time it's between your nitrogenous bases. So your adenine hooks to your thymine, your guanine is going to hook to your cytosine, again with a hydrogen bond. And that's when you have all the way down, that's what makes up the rungs of the ladder. Then once you have it, once that ladder is all put together, then the ladder kind of starts to twist, and then that's what gives that double helix image that we're so used to seeing. Now, how does this ginormous molecule carry the information that our body needs in order to function? Well, it all has to do with what order these bases are put in here. And so if you have an adenine, a guanine, a cytosine, a thymine, that is one type of instruction for the cell. And then if I would change it up and then I'd have a thymine, a thymine, a thymine, and then a cytosine, that would be a different instruction for the cell. And again, we'll talk more about that later, but the sequence of these nucleic acids is what makes it possible for all of the information that's in DNA to be able to be stored. One thing that is absolutely amazing about DNA is the quantity of information that it is able to store. I mean, if you think about the first computer that was ever invented, it took a huge, ginormous room to even contain that one computer. Now we have cell phones, which are basically just little computers that we carry around in our pockets all the time. But the amount of information that our cell phones can hold is nothing in comparison to what a DNA molecule can hold. In fact, if you have even just 1 gram of DNA, it can hold 215 million gigs of information. That is a lot of information, and again, it's just something that really testifies to the wisdom and the knowledge and the ingenuity of our Creator to be able to make something like this that holds such a huge amount of information. I'm looking forward to when we get a little bit further on in the book and we can really start talking about how this DNA starts transferring the information from one cell to another when it reproduces. So that's something to look forward to. So again, we've gone through a lot of information the past several days, and you are going to have a test over this material. I don't anticipate that I don't want you to be concerned that you have to understand absolutely everything that I've talked about, but you should know for sure the information that's on this chart that we've been filling out. You should certainly understand things about dehydration reactions and how they make bigger and bigger molecules. And so if you, as you're studying for this test, if you focus on the things that are in this table, if you focus on your own questions, if you focus on your study guide questions, you focus on the definitions that I told you that you needed to know, those things, if you know those things, that will serve you well for when you're ready to take your test.